Hey everybody, so we are talking about chapters 18 and 19, the regulation of gene expression. This is really important for cellular control and differentiation, so why we have different cells that do different jobs. And we're going to look at two general mechanisms. We're going to look at how to regulate gene expression, also how to regulate protein activity. The very first model that we're going to look at is the operon model, and this is a prokaryotic model of gene control, so bacteria. In order to understand an operon, the very first thing you have to do is take a look at its structure. So the first part is a regulatory gene, and then after the regulatory gene you have the operon area, which consists of the promoter, the operator, and then the structural genes. So here is sort of a just very general diagram. This is actually the lac operon that we're looking at. So you've got the regulatory gene, this is the promoter area, you've got your operator, and then further down you have your structural genes. So what is a regulatory gene? A regulatory gene is going to be a gene that makes a repressor protein. That repressor protein may then bind to the operator. And so this repressor protein is able to block transcription when it is bound to the operator. The promoter is going to be an attachment sequence on the DNA for RNA polymerase to start transcription. So it's where the RNA polymerase is going to bind before it transcribes genes. Then the operator is the part that sort of acts like a switch. So this is where the repressor protein is able to bind. So if the operator is blocked or the repressor protein is bound to the operator, then it's like the switch is off because it's blocked, so you can't have RNA polymerase pass, so there's no transcription. If, however, the repressor protein is not bound to the operator, then it is not blocked, and so RNA polymerase can pass through and it can transcribe the genes. So this right here is showing how the repressor protein is bound. It is blocking the RNA polymerase from transcribing the genes further on down the DNA sequence. Alright, so structural genes. These are going to be the genes that make enzymes for a metabolic pathway. In the lac operon, these genes are for digesting lactose. And so this is an inducible operon. We say it's inducible because it's only turned on when the substrate is present. So the lac operon is usually off, and then it can be induced or turned on when lactose is present and needs to be broken down. So if there is no lactose, then the repressor binds to the operator and the operon is off. So no transcription, no enzymes are being made. So this is what that looks like if lactose is absent. You don't need the enzymes down here, or these are the genes that would make the enzymes. You don't need these to be copied because there is no lactose to break down, so there's no need for the enzymes. So what happens, this is your regulatory gene. This gets copied into mRNA. The mRNA is used to make the protein. This is an active repressor protein, so it will bind to the operator. When it is bound to the operator, it blocks the RNA polymerase so that it cannot get to these structural genes. It cannot make the enzymes that's needed to break down lactose because there's no lactose to be broken down. Now, if lactose is present, then the repressor binds to lactose instead of the operator. And when that happens, the operon is turned on, transcription occurs, and the enzymes are made. Alright, so here's our whole lac operon. We have the regulatory gene. This is always going to get copied into mRNA. It's always going to make the repressor protein. Then you have here lactose. It is going to bind to that repressor protein. This repressor becomes inactive, meaning that it no longer fits, so it cannot block the RNA polymerase. So now the RNA polymerase is free to transcribe these three genes, so LAC-Z, LAC-Y, and LAC-A genes. As these genes are transcribed, mRNA is made. The mRNA is then going to be read during translation to make these enzymes, which will then be present to break down the lactose that is in the cell. So again, these enzymes digest or break down lactose. When enough of the lactose has been digested, then the repressor will again bind to the operator and it will switch the operon off again because you don't need to keep digesting lactose once it's already been digested. So the overall result is that the cell only makes the lactose digestive enzymes when the substrate is present. So it's saving time and energy by turning off the building of those digestive enzymes when there's nothing for them to digest. So if there's no lactose present, you don't need the enzymes, so you just don't make them when you don't need them. So saving time and energy. All right, our next example is the trip operon. This makes tryptophan, and this is what we call a repressible operon. So if no tryptophan is present, then the repressor protein is inactive, the operon is on, and so tryptophan is made. And this is what's considered the normal state for the cell. So we have, again, our regulatory gene, which is going to be transcribed and then translated into a repressor protein. Here the repressor is inactive, so it cannot bind to the operator. So that is going to allow the RNA polymerase to move down the DNA, and it is going to transcribe TrpE, D, C, B, and A. All right, so then you get your mRNA transcript. That mRNA transcript will be used during translation to make the polypeptides that make up the enzymes that make tryptophan. So that happens anytime that tryptophan is absent. If, however, you end up with tryptophan present, then the repressor protein becomes active. It turns the operon off. So again, no transcription, so no enzymes, which means no tryptophan is going to be made. So when tryptophan is present in the cell, again, you've got your regulatory gene that's going to make your repressor protein. Tryptophan will bind to that protein when it is present. That will then bind to the operator. It will block the RNA polymerase, and so you have no RNA made, and you are not transcribing the structural genes. So now repressible operons are examples of feedback inhibition, and so it keeps the substrate at a constant level. All right, so if you already have tryptophan, you're not going to make more because you don't need it. However, anytime you don't have any or it's low concentration, then you will be making tryptophan in order to keep that at a constant level. So your lecture question for today, I want you guys in your own words to explain to me how inducible and repressible operons are different from each other. So please make sure that you guys send me a message through the class website with how inducible and repressible operons are different from each other, and please answer in your own words. Thanks, guys.